Hello, so this Science of Sport video is related to BTEC Sport and Exercise Sciences, Unit 2 Functional Anatomy. In particular, we're looking at the muscular system and E2, the learning aim about neuromuscular process of muscle contraction. This is the second in 4 videos on this topic, and this video specifically looks at the sliding filament theory. So an impulse has travelled down a motor neuron and arrives at muscle fibres. This video looks at what happens when that impulse arrives at the muscle fibres to make them basically shorten and contract. Now, the specification basically is a list of terms that you need to be able to identify on a diagram and use as part of an explanation of the sliding filament theory. So there's actually quite a lot of names from a diagram that you need to learn. Hopefully, we'll get familiar with these as we go through. I'd like to start by showing a short video to illustrate the sliding filament theory, and it also again shows you some of this terminology on diagrams.
We just learned about the structure of muscle, like skeletal muscle, which is what allows you to move your body around. But how exactly does this work? If you decide to lift your arms up in the air, what is happening on the molecular level that produces this motion? It's not magic, it's just a staggering amount of chemistry. So let's dive back down into those muscle fibers and get a closer look. As we recall, any skeletal muscle is made of fascicles, and each fascicle is made of muscle fibers, which are the individual multinucleated muscle cells. From there, we can zoom in farther on one of these myofibrils, which are in turn comprised of myofilaments arranged into sarcomeres. And this is the contractile unit, the one that lets muscles do what they do. Now let's get a closer look at these structures, because this is where all the action happens. Looking at a sarcomere, we can label a few regions. We can see these darker A bands and the lighter I bands, which are aligned and responsible for the striated appearance of skeletal muscle. Within each A band, we can find a lighter region called the H zone, and this H zone is split down the middle by something called the M line, made of a protein called myosin. The I bands are also split down the middle by a region called the Z disc. We can identify an individual sarcomere as being a section from one Z disc to the next, and this is the functional unit of skeletal muscle. If we zoom in even more, we can see the myofilaments that make up these regions. As we discussed previously, the thick filaments contain myosin, and these extend across the A band, connected at the M line. The thin filaments contain actin, extending across the I band and into the A band. There are also elastic filaments made of titin, spanning from the Z discs to the thick filament, and then continuing on, acting as the core of the thick filament. Now let's zoom in even further on these two main types of filaments. Myosin is a protein with 2 globular heads pointing outwards and a long tail, which contributes to the structure of the filament. The heads are the site of all the activity, since there are ATP binding sites, as well as actin binding sites, meaning this is where the thick and thin filaments will interact by making cross bridges. It's important to note the lack of myosin heads in the center of the sarcomere, just as there also are no thin filaments. Speaking of those, as we said, they are made predominantly of actin, and 2 actin filaments will twist together to form the backbone of the thin filament. Each actin subunit has an active site where myosin can bind, and when a muscle fiber is relaxed, these are blocked by spiraling strands of tropomyosin. There is also troponin, a globular complex of 3 polypeptides, one of which binds to actin, one of which binds to tropomyosin, and one of which binds calcium.
So that video clip really introduced the nuts and bolts of things, the strands, the myofilaments that make up a muscle fiber or a myofibril. Let's try and put that together with some slides or some diagrams again. So remember, we've got our whole muscle fiber. This is a bundle, a fasciculi or fascicle, a bundle of fibers. This pulled out here is a single muscle fiber or myofiber. The single muscle fiber is made out of lots of myofibrils. So this is a myofibril. And we've taken the single myofibril and represented it here in a slightly larger version. What we can see already is that the myofibril shows the stripes, the striations that you might see if you looked at muscle fibers under a microscope. And this myofibril is made up of two types of myofilament. Thick myofilaments, in this diagram it's blue, these are called myosin, and thin myofilaments, these are called actin. And a simple way of remembering the difference between the two is actin thin, and then the myosin are the thick ones, so actin thin. Here's exactly the same diagram, but a little bit larger and hopefully clearer for you to see. So remember, this is the myofibril, and lots of these myofibrils make up a muscle fiber. Okay, and we've got our different colours, our striations looking under a microscope. Now, importantly, language we need to recognise is what is the terminology that describes all the different parts of a sarcomere. This single unit of a myofibril here is a sarcomere. The sarcomere goes between Z disc and Z disc. Okay, so this is one sarcomere. Another one would be there, another one would be there, and so on and so on, so on, all the way down along this myofibril. The sliding filament theory, so remember these actin and myosin are filaments, myofilaments. Sliding filament theory talks about these filaments sliding in between each other. And when that happens, these Z discs move towards each other, get closer together, i.e. this sarcomere shortens. Now if you imagine this sarcomere shortening here, 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 all the way along a myofibril, you might understand how a muscle shortens when it contracts, and we'll look at that again in more detail in a moment. So anatomy, a sarcomere is the contractile unit of a muscle fiber, and it's between the Z discs. What we've also got then is the blue myosin filaments. We've got in this diagram, pink actin filaments. So the actin filaments are actually attached to the Z discs. The actin filaments attached to the Z discs. The myosin filaments aren't. The myosin filaments sort of have a central line, an M line. Where you look down a microscope and you just see myosin, so this bit of blue in the middle is just myosin, that's the H band. Now, if you imagine the pink filaments getting pulled towards the center, that H band would get smaller and smaller and smaller. Anyway, the myosin only is the H band. The A band is basically the length of the myosin. So the blue to the blue, the end of the blue to the end of the blue. We've got the I band. So the I band is where there's just actin. So the H band is where there's just myosin, and the I band is where there's just actin. And you can see it's from basically the end of the blue there, it's just actin. Get to the Z line, it's still just actin. Get to the next blue, myosin. So that whole gap either side of the Z disc makes up the I band. And again, during contraction, that area, that sort of length there would get shorter and shorter when the sarcomeres contract. So some basic anatomy that you need to get your head round and be able to label on a diagram. I'm going to show you lots of different diagrams of the same thing, hoping it'll stick for you. So this is a more factual diagrammatic version, but it's the same. Okay, Z line, M line is the middle, H band is myosin only, I band is just actin, and this shows any overlap there is. Okay, the A band is the length of the myosin, and that's just a cross-sectional view of it. This slide shows actually a microscopic version of the same. So again, we've talked a lot about striations. You can see this muscle fibre, it clearly looks striped under a microscope. And if we take out that little segment there, we get this right-hand image, and this shows our labelled sarcomere. So the sarcomere goes from this Z line to this Z line. Here's the myosin only in the middle. Here's the actin only either side the Z band. This is the length of the myosin, which is the A band, and the gap in the middle is the H band. We said that already. Okay, so just a slightly different perspective. Now, what the video also showed you was more specifically, what does each myofilament look like? The thick myosin filaments have those arms and the heads. The actin-thin filaments are strands of globular proteins. Okay, so we've got them made up of little blobby bits basically, and the myosin have the arms and the heads. Slightly bigger version of this, so this would be our actin, strands of globular proteins. This is our myosin with the myosin arm and the myosin head, all really important for how sliding filament works. So what I suggest you do is just pause the video. This information just reiterates what I've just explained to you. Make the connection if you can. Actin are the thin ones, myosin have the arms, and digest so that you could recognize these on diagrams. So let's just go back a step and paint the picture of the story so far. We know an impulse comes down a motor neuron, gets to this motor end plate, it's got to get across the synapse. We've got this ACH, acetylcholine, that is the neurotransmitter that gets the impulse, the action potential across the synapse into the receptor sites. And what we're going to look at through sliding filament theory is what happens next. How do we get from an impulse arriving here to these sarcomeres along the length of these myofibrils and these muscle fibers shortening? So arriving at the receptor sites here, you can see these arrows passing along the top of the sarcolemma. The sarcolemma is the sort of outer sheath of this muscle fiber. So the impulse actually spreads along the sarcolemma, but also what I want you to recognize are these tubes, these tunnels that go down into the muscle fiber and in between all these myofibrils. These are called T-tubules, so it's pretty easy to remember. These T-tubules are tubes that spread the impulse down and in and throughout the muscle fiber. Importantly, what they do is they trigger the release of calcium from this area here. This is called the sarcoplasmic reticulum. Basically, it's where we store calcium in our muscles. So an impulse comes along, spreads into and down through the muscle fibers via T-tubules, triggers the release of calcium from the sarcoplasmic reticulum. And what we need to understand, and we'll look at in a moment, is why is this calcium so important? Why is it released? How does it help us make sliding filament happen and make muscles shorten and contract? Just again, quickly pause the video. This is similar or the same information I've just explained on a previous slide, but you might want to see it in writing, so make sure you take time to digest this bit of muscle cell anatomy. So the question is, why is calcium so important within the sliding filament theory in terms of allowing our muscle cells to actually shorten and contract? We've got an impulse to the muscle cell, it's gone down the T-tubules, it's triggered the release of calcium from the sarcoplasmic reticulum. We kind of want to know, so what? How does that help? Well, it's really important in enabling a cross bridge to form between myosin and actin. Now, if you remember, myosin have the arms and the head, and actin are those strands of globular blobs of protein. When our muscles contract, basically the myosin head attaches to the actin, and it attaches to a special site called the actin binding site or myosin binding site. It's where they can connect. So this slide actually shows our actin filament. So these strands of blobs of actin, and what we also can see are these little, can you see that it's slightly covered there, it's available there, it's more visible there. These are the places, the binding sites, where the myosin arm can attach to make a cross bridge. Now in this first image at the top there, these binding sites are blocked by tropomyosin, this strand wrapping around. Can you see it's blocking all those binding sites? And while those binding sites are blocked, a cross bridge cannot form, myosin can't attach to actin, and we can't contract our muscles. So in order to contract our muscles, we need to make these binding sites available so that the myosin head can connect and a cross bridge can form. So this ultimately is where calcium plays a really important role in enabling contraction to happen. What the calcium does, when it's released from the sarcoplasmic reticulum, when an impulse has come, remember an impulse is this action potential that's an instruction to hopefully innovate muscle contraction, make muscle contraction happen. And when it arrives at the muscle cell, the impulse triggers the release of calcium from the sarcoplasmic reticulum. The calcium, as you can see here, binds with this troponin complex or troponin. So the complex is there when it's blocked, and the green blobs of calcium combine with the troponin complex. Now what it does, it basically makes that troponin or troponin change shape, change form, and moves. But when the troponin moves, it's also connected to and pulls the tropomyosin, this grey strand, out of the way. It pulls it with it, changes shape with it. So the calcium binding to the troponin actually has the knock-on effect of moving this tropomyosin out of the way and then ultimately making this binding site available. And while this impulse is present, the binding sites can be used by the myosin heads. So we now need to look at, okay, what happens when a myosin head attaches to a binding site and forms a cross bridge? So what?
Speaking of those, as we said, they are made predominantly of actin, and two actin filaments will twist together to form the backbone of the thin filament. Each actin subunit has an active site where myosin can bind, and when a muscle fiber is relaxed, these are blocked by spiraling strands of tropomyosin. There is also troponin, a globular complex of 3 polypeptides, one of which binds to actin, one of which binds to tropomyosin, and one of which binds calcium. Beyond this, we must be aware of the sarcoplasmic reticulum, a series of tubules that surround each myofibril. These regulate levels of calcium, which is needed for muscle contraction through storage and release. This structure includes the T-tubules, which sit at each A-band, I-band junction, encircling each sarcomere and helping signals reach every region of the muscle cell. Now that we've examined each structural component, let's describe how everything works using the sliding filament model of contraction. This says that when the nervous system stimulates muscle fibers, the myosin heads on the thick filaments will interact with the binding sites on the actin subunits. These attachments will form and break several times as the thick filaments pull the thin filaments in towards the center of the sarcomere, thus pulling the Z discs towards the M line. The I bands shorten and the H zone disappears.
Now that video clip actually showed you sort of two processes. It showed the diagram at the top with the sarcomere sliding in between each other or overlapping, and it also showed you the process with the myosin head and the actin forming cross bridges and breaking cross bridges and forming cross bridges repeatedly on the bottom. So I'd maybe recommend re-watching that a couple of times. But the thing you need to remember is that the myosin arms on the thick filaments pull the Z discs closer to each other. So this diagram actually represents that through this sort of tug of war analogy. The blue sort of athletes here are pulling on the actin. They've connected, formed a cross bridge. They're pulling this actin myofilament, which is attached to the Z discs, and moving the Z discs towards the middle. Similarly, these tug of war athletes on this side are doing the same, but pulling this Z disc again towards the middle, the M line in the middle of the sarcomere. So we've got pulling in both directions, ultimately making the Z disc get closer together and closer together and closer together. This diagram shows it in a slightly more formal way. So this would be when there's no impulse. This is a relaxed muscle fiber, a relaxed sarcomere. We've got our light I bands, we've got our H zone in the middle, which is just the myosin. This diagram represents a contracted sarcomere. So the myosin blue arms here have pulled the actin and pulled the Z lines in. You can see the Z line's gone from there into there and from there into there. And there's much more overlap. And in fact, there's no H zone at all to be visible. The I band has got smaller where everything's sort of been concertinaed and overlapped. So sliding filaments actually make sense. These two types of filaments slide between each other. I'm really keen where I can to show you anatomical images. So you'll see a lot of these kinds of diagrams, relaxed, partially contracted, fully contracted, and how the sarcomeres change in length. But this shows it anatomically. So this would be your microscopic version of it. So this is the relaxed sarcomere, and this is the contracted sarcomere. We just need to add a little bit more detail. So we've talked about our actin have special binding sites that are hidden by this tropomyosin strand that wraps around the actin. It's only when calcium appears that and binds with this complex, this troponin or troponin complex, it pulls the tropomyosin strand out of the way and makes the binding site available. What that means then is that our myosin arm or our myosin head can form a cross bridge, can connect. You can just about see the sort of darker binding sites on that actin strand there. So this is a cross bridge formed, and then what happens is the myosin arm pulls the actin along over it, slides between it. One thing we haven't mentioned so far is this is an active process. This is a dynamic, energized process. And because the actin arm has to pull the actin and actively sort of move it along, it needs energy to do so. And you will need to understand that energy comes from this special compound called ATP, adenosine triphosphate. Now you'll learn much more about it in the physiology unit, but we're just going to mention it here. Basically, it's an adenosine and 3 triphosphates bonded together. When it splits and splits off a phosphate like this, it releases energy. And that energy allows the cross bridge to form. And that energy allows what we call the power stroke, the myosin arm to pull on the actin. Now, we need ATP to bind and pull, but we also need a new ATP molecule to allow the release of this arm. So we need ATP a lot. Without ATP, we cannot make muscles contract. This process can't happen without ATP. So ATP is needed, or the splitting of ATP is needed to bind and to power stroke, to pull, but also to release as well. So from this cross-bridge formation here, another ATP would attach. That's what this diagram relaxes, and it moves away. Now, I appreciate very much this topic is quite involved. You will need to pause the video and take in this information. I'm not going to go through this particular slide. This is text version of what I've explained on some of the previous diagrams. Last couple of bits. So again, pause the video and just work through these two diagrammatic versions of the explanation of sliding filament theory. And I think this is quite a nice summary slide. The impulse arriving, getting across the neuromuscular junction, the impulse spreading down the T tubules and into the muscle fibers, calcium released from the sarcoplasmic reticulum, calcium binding with troponin to release the binding sites, a cross bridge forming with the myosin arm, the power stroke pulling the actin and the Z lines towards the middle of the sarcomere, and that works, remember, in both directions. In both directions. So that's of course the description of how we instruct and send impulses and action potential to make our muscles contract. On the right hand side here is the explanation of when we want to stop muscles contracting, what happens? Well, basically it's everything in the sort of negative version. So the impulse, there's no impulse. So here we had an impulse to make it happen. Here the impulse is ceased or stopped. There's no ACH therefore released to pass on any impulse. There's nothing going down the T tubules. The calcium goes back into the sarcoplasmic reticulum. We don't have a cross bridge. The binding sites are covered again by the tropomyosin, and our sarcomere is in its relaxed state. So there's no cross bridge forming and no myosin arms pulling on the actin. So it's a reasonably complicated process that you need to be able to explain. I just want to finish by showing you two exam type questions you may come across. So this one is more of a know the parts. So it's given you a diagram, it's asked you to label the two identified anatomical features of the sarcomere, and hopefully you would have recognized that A was the actin and B was the Z line. But the other type of question you might have is more of a process one. So here for 8 marks, it tells you the sliding filament theory accounts for the process of muscle contraction. It shows you the diagram, so it gives you some clues. It shows you the relaxed and the contracted version, but what it wants you to do is analyze how this process happens. How do we have sarcomeres that shorten? And so the mark scheme, again, please pause the video. I'm not going to go through it bit by bit, but the mark scheme is here, and it talks you through the story or the process of sliding filament theory.